Good morning. Danielle Verna here, project manager for the Scientific Advisory Committee, and I am joined by Dr. Morgan Bender of Fjord and Fish Sciences. This item on the agenda is an action item. We're asking for board approval of 3 reports associated with long-term environmental monitoring. Morgan's going to go into detail on the sampling that we did in 2024 and the results of that sampling. I just wanted to point out something different this year. There are 3 reports associated with LTEMP. So there's the pretty standard for the past few years summary report, and then the technical supplement, which goes into a lot more detail on the results. And then new this year is a report on some sampling we did of metals in sediment outside of the Valdez Marine Terminal and at Gold Creek. That was a preliminary study that was kind of the extension of the report that was presented a couple of years ago from Dr. David Podgorski. He had taken some samples from the ballast water treatment facility and identified some metals in the effluent from the treatment facility. And SAC decided that we would like to take some samples of sediment to see if there was accumulation of metals in that sediment. So just a preliminary study, and Morgan will share the results of that. And that's it. That's your...
Great. Thank you, Danielle. Happy to be here in person this year. My name is Morgan Bender, and I'm going to talk about the Long-Term Environmental Monitoring Program, or LTEMP, and what we did in 2024. Review that program briefly, talk about the methods that we used, present the findings. It's the 31st year of LTEMP, so we have quite a long time series to talk about. And then, as Danielle mentioned, a pilot study on the sediment metals. Here's a timeline. It has quite a few points on it, and I will already give a plug for a poster that's in this room in the corner right there, which has a similar timeline. So if you want to spend some time studying it later. Generally, the program started in 1993 after OPA 90 and the formation of the council. Some notable events are spills that we see in our data set at the terminal in the purple drop, as well as at Gold Creek. LTEMP went through a sampling reduction in 2010 so that we took fewer samples and were able to focus more specifically on our known reference sites and around the active areas such as the terminal. We added seawater sampling via passive samplers in 2016. 2020 is when the muscle transcriptomics, or small biological changes in mussels that were exposed to crude oil, was added. In 2023 was the 30-year anniversary of LTEMP, and now in 2024 we did a pilot study on sediment metals. So lots of changes over the time and a few things happening in the environment. We have about 245,000 chemical data points in the data set, so there are lots of stories to explore chemically, and I'll just present a few of those as they would relate to environmental health. In 2024, Danielle, Jeremy, and I sampled around Port Valdez and in the North Gulf Coast at the stations that I'll show in the map following. The chemical analysis was done by state-of-the-art laboratories in the United States, in Massachusetts and in Oregon at the Food Security and Environmental Stewardship Laboratory. And then, as Danielle mentioned, the reporting was divided up into 2 reports for the standard LTEMP. We have a summary report, which is more of an executive summary, hopefully accessible for everyone who's interested, as well as a really meaty technical supplement. And that's been the same reporting strategy that we've used for the past few years since I started with LTEMP in 2021. So we have a pretty dialed format for those reports. And then additionally, we have the sediment metals report for this year. And then here are some glamour shots from our field work in Port Valdez. Danielle and I in the upper right-hand corner working with the passive sampling devices, and then all of us smiling around our Von Veen grab, which takes a nice undisturbed sample of the subtidal marine sediments so that we can understand what those benthic organisms are experiencing. Here's a map of our sampling sites. Most of it is focused in Port Valdez now. We take samples around the terminal. Right outside the effluent from the ballast water treatment plant is where we take our sediment sample. So it's this yellow dot here. It's nearly invisible. So good thing Joe has glasses. And then we also take a sediment site over at Gold Creek, and this is our standard reference site in Port Valdez. Additionally, in 2024, we sampled the sites that we missed in the extended sampling of last year, where we look at a lot of these previously Exxon Valdez oiled sites. So our blue mussel samples were taken around the terminal at Gold Creek and at the Valdez Small Boat Harbor, as well as these North Gulf Coast samples sites in Ayalik Bay, in Windy Bay, and then also in Chugach Harbor in the Kodiak Islands. The passive sampling devices, so this samples the seawater, were taken around the terminal, again at Saw Island and Jackson Point, as well as our reference site at Gold Creek. One of the questions that came from SAC this year was to really dive into the site selection criteria. LTEMP was started over 30 years ago, so some of us may have forgotten why we chose these sites. So we dug into literature and we found the real reason for these sites being chosen. So they divide up into 3 categories. One is current oil and terminal and tanker activities. So that's the loading sites around the terminal, anchoring like at Knowles Head, and the transportation routes such as the Zykoff Bay site in Hitchenbrook Entrance. The previously oiled sites, so we have all of our spill-affected downstream areas that move into the North Gulf Coast, as well as reference locations. So we have a reference location in Port Valdez, that's Gold Creek. We have a reference location in Prince William Sound, that's Sheep Bay outside of Cordova. And we also have a reference location in the North Gulf Coast, that's Ayalik Bay. So if we color code those, you'll see that there's a spread for each one of our sampling matrices. That's like just a thing that we sample. So sediments is a matrice, blue mussels, another matrice, and the seawater via passive sampling device. So you'll see that this year we sampled only 2 of those historically oiled sites in the North Gulf Coast. But those are normally sampled every 5 years. And because we missed them last year, we got to visit them this year. And here's some only slightly glitchy. I aim to be less glitchy than I was last year. Here's a flyby from Windy Bay. So our sampling site is coming into view now. We had a really nice time sampling out of Homer via float plane. In the picture up to the upper right, we have Jeremy cleaning some of these low-density polyethylene strips or passive sampling devices. We have the fishing vessel Equinox that we used as our sampling platform to take sediment samples and to launch from to get our blue mussel samples. Danielle taking meticulous notes and preparing our sampling gear, as well as a scenic flyover of Ayalik Glacier in Ayalik Bay on our way to Windy Bay. Here we're going to jump into the chemical results. This is presenting the sediment data from Valdez Marine Terminal in purple and Gold Creek in gold. The figure plots a concentration of toxic hydrocarbons in parts per billion. And then we've got our time scale on the lower x-axis. And what we see is quite a bit of variability during the first 15 years or so of sampling. And then since then, we've got pretty low and maintains concentrations, really not a lot of difference between our 2 sites in Port Valdez. 2024 results are presented all the way to the left in the bar plot, and we have about 150 parts per billion of these toxic hydrocarbons at the marine terminal, and a 5th of that at Gold Creek. When we look at where these hydrocarbons are coming from, we use a few different techniques, and one is to look for a tight match between the Alaska North Slope crude that flows through the pipeline, and some of which is processed at the ballast water treatment plant. And we do see that the terminal sediments have a matching and strong signal from an Alaska North Slope crude. But we also see some other sources, likely of combustion related source. So whether that's exhaust or some faraway sources, unidentifiable through my level of analysis exactly what that is, but things in addition to Alaska North Slope crude. So that's what we see at the terminal. If we look at what we see from Gold Creek, we do not see the same Alaska North Slope crude signal. We see more of a mix, either from runoff from Gold Creek itself or other sources in the area, both pyrogenic of combustion origin, whether that's exhaust or forest fires, but also some petrogenic. There are local natural petrogenic sources that are not Alaska North Slope crude, so it's possible that that's what we're seeing here. If we look in the tissues, this is the same style of representation for the toxic hydrocarbons, the polycyclic aromatic hydrocarbons, and we're still in parts per billion here. We see our highest levels at the Valdez Small Boat Harbor, and this has been consistent over the time we've been sampling this site. I like to think of it as a sort of positive control for, yes, these guys do take up hydrocarbons, and when they're in a highly disturbed area like the Valdez Small Boat Harbor, where there's lots of boat traffic, there's lots of little spills happening, these guys do respond. We also saw levels at Windy Bay, Ayalik Bay, and Shugak Harbor. These are our remote sites in the North Gulf Coast and on Kodiak. We did see quite a bit of variability in these samples. And then our sites around the terminal, Jackson Point, and the terminal itself were pretty closely related with some variability, and our lowest levels were seen at Gold Creek. If we look into the forensics of these guys, we see a lot of mixed sources, clearly pyrogenic, so that's your exhaust or your forest fires or your other combusted hydrocarbons. But we also see some heavily weathered crude oil sources in our Windy Bay, Ayalik Bay, and Shugak Harbor sites, but no strong Alaska North Slope crude. One thing that we did observe more on the biological side is that the robust mussel beds, which were originally chosen to be good sites to sample, these mussels naturally collect what is in the water column indiscriminately based off how they feed. So they're used worldwide as an indicator or sentinel species looking at water quality. So our sites were originally specifically selected because there were robust mussel beds there. And when we finally made it to Windy Bay, we did not find the robust mussel beds that once were. So they were actually probably predated pretty strongly by these intertidal sea stars. So that was an interesting biological observation that we made, which could probably affect some of the chemistry that we see in that area. So the variability that we saw at this site might be because these guys are already under some other stressors. So that's a reminder too that when you have these long-term data sets, you really need to consider other factors that are influencing the biology of these organisms. Great. Here is the time trend for the mussel data. Each one of these dots is a individual mussel replicate that we've taken over time. So you can see how much of a cloud we have already made. I think that's a testament to this group and their commitment to independent, robust environmental monitoring. Thank you for that. The colors of each one of these dots is those three sampling groups that I mentioned earlier. So the red ones are the active areas. The green ones are the oiled sites from the Exxon Valdez, and the blue are the reference. So if we look at the trend lines for this, we see that that high variability time, similar to what we saw in the sediments, we have a period of low hydrocarbon levels. And this is exactly where we reduced our sampling scheme and then slightly elevated from there. And I know probably on everyone's mind when I talk about hydrocarbon levels is what does it mean for the biology, and we're going to get to that. But first I have to pound these numbers in. This is a green line. Sorry, it doesn't show up so well here. And we can see it's pretty similar. Started out even lower, which is maybe an interesting observation that is not part of this year's analysis, but could be next year's. And we do see consistently low levels in these previously oiled sites. And then if we plot the reference over top, and I've taken out our positive control from this particular trend line, we see pretty similar here. So it does play out in these most recent years as I guess we would expect that the areas that are under active terminal and tanker operations do have slightly elevated levels compared to our previously oiled and reference sites. And this is just the mussels that we're talking about here. Lastly, what's happening in the seawater? So the way we sample the seawater through the passive sampling devices gives us an idea of what compounds are bioavailable to animals like fish. So we're looking at what dissolved hydrocarbons there are. This is different than what you'd find in the sediments and even what you find in the mussels because they feed, filter feeding, they can take in things like oil droplets that maybe wouldn't affect pelagic organisms in the same way. So we see quite a bit of variability from Gold Creek. This is actually in parts per trillion of our toxic hydrocarbons. So now we're at really, really low detection levels. And we see a lot of variability at Gold Creek. That's likely because there's some runoff from a stream there, and we have a lot of small hydrocarbons in this particular sample. And then, really tight, low levels at the terminal and Jackson Point. The historical time trend for these passive sampling devices since 2016 looks like this. We do see a nice response from that 2020 terminal oil spill, and then the 2024 results fall within the trend that we've seen. So nothing super surprising. That variability at Gold Creek can be explained away when we start to dig into some of the chemistry more analyte specific or looking at single compounds as they vary. But what do these really mean for aquatic life in Prince William Sound? So if we look at the sediments alone and look at the individual compounds that were measured, we see that there is low risk. It's about 1% of the EPA benchmark threshold for chronic effects in benthic organisms, and that's at both stations. If we look at our intertidal mussels, I categorize it as low risk with variability. The mussel hydrocarbons are likely too low to elicit adverse effects in mussels. Some of these higher levels that we see in mussels around the small boat harbor, for example, those mussels might be slightly affected. Maybe they grow a little slower, and that's where the biotransfer omics analysis will really reveal what could be happening in those mussels. In the passive sampling devices, looking at seawater, we see low risk. There's not a lot that could come in contact with the biology. The water-soluble hydrocarbon concentrations are likely too low, even for sensitive life stages like embryos and larvae of fish, which is where a lot of this research is at the moment. And here is the preliminary results from the pilot study on metal accumulation in the sediments. Again, the purple bars are from the terminal and the yellow is Gold Creek. Each one of these panels, the A, B, and C, is metals that have a widely different concentration group. So that the numbers here on the y-axis are pretty different. Here we're in the 40,000. Here we only go up to 90. And this panel is actually just all estimated values because they're below the reporting limit for that specific metal. What we see is that there are metals present in our sediments. That's not really surprising. We're in the parts per million here of metals. We see quite a bit of iron, aluminum, magnesium, sodium, calcium, potassium. I'm not going to read them all, don't worry. The terminal sediments did generally have higher metal concentrations than Gold Creek. When I ran some statistics on this, I found that it was significantly higher for quite a few metals, which are listed there. Iron, zinc, copper, chromium, and vanadium. But what would this mean for the biology, you might ask? Me too, as a biologist. So that is in our next slide. This is the metals that's presented in a slightly different way. Here I've compared each one of those samples to the National Sediment Quality Threshold for a low-level effect on organisms. So essentially what we see popping up above this line in the middle is metals that exceeded this threshold where we would expect more than 10% of an exposure to lead to an adverse effect on, let's say, a worm or a clam. The guys that are below this threshold are, nationally speaking, not of an ecological concern. So we do have quite a few metals that pop above this line that are in this, red zone. But what I'd like to draw your attention to is that it is apparently a regional thing. So each one of our samples, both the terminal and Gold Creek, exceeded this threshold for potential negative effects. We do have quite a few metals that maybe could be investigated further. Iron, vanadium is something that's involved in steel production, so you do find it around vessels. But because we have, somewhat similar, although they are statistically different results between our two sites, it's not that it is exceeding this limit only at the terminal and not at Gold Creek. So I think we are interested in exploring this metals data further. Surprisingly, we didn't find a lot of metals data from the Port Valdez area. So if anybody knows of metal studies that have happened, I'd be happy to compare it to our results. But at this point, we need to figure out what to do with our information. Great, moving forward. As a true scientist, I'm always saying we can do more, so consider expanding sampling efforts, things like adding more recent sources to our source catalog. So that's maybe understanding really what's coming out of Gold Creek or what is going on in the small boat harbor that leads to these mussels having consistently elevated hydrocarbons, maybe getting a recent sample from the ballast water treatment plant. Increasing the project visibility, so expand the dissemination. Maybe I just wanted to add that word to our presentation. I feel like you have to say that. One of our efforts is already going to happen. It's this poster that you see on the wall. So I'll be presenting that next week at the Alaska Marine Science Symposium. And the idea by bringing it out to other scientists is that we might have people that are interested in that data set that could use it to dig deeper into hydrocarbon effects on organisms, the weathering and fate of these compounds in the environment, or maybe they can also, run parallels with their own study. And Danielle and I are still working on making the data set more accessible to people outside the council. It is available on the website, but finding places where other data lives so that this data can be curated there. And lastly, evaluate specific aspects of LTEMP. The Scientific Advisory Council is always reviewing the LTEMP findings in great detail. So, considering the changes that we saw at the Windy Bay mussel community, maybe that's a site that needs to be evaluated, as well as investigating further the metals data that we have and discussing how to move forward. As summary, so we do see the consistent trend in the forensics of where these hydrocarbons are coming from. The site that is directly outside the terminal does reveal Alaska North Slope crude signatures, whereas all of the other sites that we sampled have more mixed sources. We see low potential environmental and toxicological risk posed by the hydrocarbons. This study almost entirely focuses on hydrocarbons, though we do analyze a few more things in the samples, but we are focused on hydrocarbons here. Historical trends still reveal low concentrations that spike after spills. And our pilot study did find metals accumulating in Port Valdez sediments. And that is all I have for you guys today. Thank you so much for having me.
Thank you, Dr. Bender. That was an extensive report. Thank you for that. Are there questions? Dave Jenkins.
Thank you. Yeah, great, great report. I was, I can't, all the times I did it, but I can't remember early on, did you run across any actual sampling of the EVOS lingering oil that RCAC did, and chemical analysis of the lingering oil itself? Did you run across any of that?
I do believe there are some of those samples in the data set. I haven't dug in those specific samples because I am always trying to say, okay, what's the most consistent comparison that I can make to what we've been doing in 2024 or the previous 4 years that I've done this study. But I do know that those are in the data set. One direction that I'd like to go, though, time is always limited, is to try to tie the LTEMP results to some of the other work that's going on, for example, DEC's lingering oil project, to see if we can find some parallels between what we're finding in the mussels. Unfortunately, the mussels are, living, I'm not going to say breathing because they're mollusks, but they don't stick around the whole time. It's not the same as going into the sediments where you have a nice log layer. Like these
Sure.
guys are respiring and exchanging
Right.
their compounds. So because we take mussels from the lingering oil sites, I really wouldn't expect to see that, long-term trend. I'd expect to see what they've been experiencing in the past few months.
I recently had an inquiry from Gary Shiganako, who's recently retired with NOAA in Seattle, about any recent chemistry on the lingering oil. I think mostly to look at degradation over time. And then Ron Heinz's survey in 2020, at least his report 2023, they took samples, but then just put them in the freezer. They did not analyze them. I don't know why, but probably cost of that. But anyway, Gary was very interested in that. Was wondering if can help with any of that, or I suppose he should contact Heinz about figuring out how to analyze some of that.
Yeah, if there's things in the freezer. To my knowledge, probably the last 10 years at least, I could speak with some confidence, we do not have a sediment sample from a site with historically
Very
oiled
early
site.
on that I think.
Yeah,
some
early on, I think we were taking samples from wherever we could find
Right. Okay, thank you.
good information. Thank you, Dave.
Steve Lewis.
Yes, thank you, Mr. President. Dr. Bender, you mentioned ANS, and we know over the 30 years of the period here, the composition of the crude stream has changed. I'm not sure how significant that is because I haven't looked at the data myself recently. But my question is, does that affect your sampling, the change in the composition? And if so, how do you compensate for that changing signature that one would expect from the changed composition of the stream?
Great question. I'm happy to have an answer for that one. We do take recent samples from the Alaska North Slope crude so that we can trace those changes in the chemical composition. I think our most recent sample is from about 4 years ago, so we do have to take some assumptions that what we're seeing in the sediments is related to that 4-year-old sample. We take the top layer of the sediments as well, so we hope we're comparing recent to recent. The way that the forensics technique was developed is that it looks at these petrogeochemical recalcitrant biomarkers, so compounds that don't break down in weather in the same way that a hydrocarbon would. So they're more of a line that we can follow from the crude oil to the sediments. So recent samples and recent sediments.
Okay, thank you.
No, thank you.
Thanks. Yeah, Steve kind of touched on my first question, was just if you kind of knew the profile of the crude over time, like each year it's a little bit different based on what's coming out. Could you theoretically find out, oh, this is like if you took a sample, this is lingering from, and then the time period?
Yeah,
of it that-
definitely.
okay,
Yep, you can totally do that.
Awesome.
Yeah, you can even give a sort of a percentage contribution of specific types of oil. So there are some pretty advanced statistics because LTEMP is something that is analyzed at this point every year. And we're interested in just a few questions. There's really sort of scratching the surface level for each compound or each sample that we take. There's about 160 compounds that are analyzed. So the results that I presented here, I'm embarrassed to say only about 42 compounds were represented. So there's really a lot we can tell from this data set. And so that's part of our idea with making the data set more visible, is that people who really want to get into the weeds on it can do that and save your guys this time for those of you who are more interested in, you know, what it means for fish or the ecosystem health.
Great. And then my last question was around the sediment samples. Did you guys take a kind of like a general soil profile? I know you mentioned that there was some higher levels that are kind of dangerous, or so to speak, for biological samples, but it was common amongst different sites. So I was just wondering, is there's like a general metal soil profile? Because maybe it's just common in the area to have higher levels of toxic metal or different types of metals.
I'm not sure I understand your question, but the idea of like a common layer or a common concentration is, I think, maybe baseline. Is that what we're trying to talk about?
Yeah.
Okay,
So just
so-
looking at like iron, this particular area has this percentage of iron.
Yeah,
But
I
across multiple
I think
metals.
being in a heavily glacially influenced area like Port Valdez or all of Prince William Sound, you're going to see a lot of ground up rock in your water and sediment. And so that's where a lot of those metals are coming from directly. So I think we're living in a very metal rich water. And that's where the national sediment guidelines for, you know, protecting benthic life, it might not be as applicable to Port Valdez as we'd like. Because those organisms, if they chose to live there, they're already going to be in a really metal rich environment.
So it'd be harder to tie it to the actual industry.
Yeah, we would definitely have to dig more into the literature to see if it's really true that those organisms are experiencing some adverse effects from the elevated metal concentrations that we found. Or maybe we could run some experiments, but we'd definitely start out by doing an extensive literature review, and maybe not just relying on the national metal guidelines.
Great, thank you.
Jim Herbert.
Thank you, Dr. Bender. Good report, nice graphics. I was curious, if I couldn't quite pick it up from your description, does the fingerprint of any of those hydrocarbons match historic Exxon Valdez oil?
What we'd want to do is find a source that was of similar age. So looking at what state Exxon Valdez crude is it now in the environment. And most of what I've read lately, and DEC is working on this pretty intensely right now on their lingering oil project, is that that oil is not bioavailable. So it means it's kind of capped and not really influencing the surrounding ecosystem. So you wouldn't find those same compounds in a mussel that's even kind of closely related to a site that still has oil present at depth. It could be done, I believe, using those recalcitrant biomarkers, the ones that don't break down in the same way that hydrocarbons do, but because of the extensive weathering that the Exxon Valdez crude has gone under over the past 35, 6 years, it looks pretty different now. And I think it will persist with just sort of small incremental changes. I think they say 1% to 2% change per year for the next few decades in the places that it is.
I always had the impression that some of that weathered oil in the sediments, and Dave could sure point you to where they are, you probably know yourself. It's a little bit like lead in paints. If you don't disturb it, it's okay. But if you start scraping and sanding, and then you're potentially inhaling the lead. So possibly then that Windy Bay, where it's exposed over the years, has more wave action. Maybe that puts that stuff in the environment.
Yeah.
Two other things quickly. I always wonder about mussels, and I think you said it a few minutes ago. It's basically giving you a snapshot of what's been happening in the water column over the past few months,
Correct.
because they purge themselves, and so that's kind of a snapshot. So if it's a 5-year or a long gap, we could be missing vents.
Yeah.
And I like what Ben was saying, that whole geology of that region may indicate that these metals that you detected are just the normal background giving glaciers and the newness of mountains. So it'd be nice to maybe just do a metal thing elsewhere just to see what the background is.
Yeah.
Thanks.
Wayne Donaldson.
I don't have any questions. I just wanted to offer my congratulations to Dr. Bender and Dr. Verna. This was an excellent report. I've been reading LTEMP reports for 10 years, and this is by far the best, most easily put together report. They're very, I found them very open to suggestions for changing the report from prior authors and prior PIs. So I just wanted to publicly acknowledge the effort that they put in. Thank you.
Yeah, I agree. Very well done. Thank you. I had been involved with NOAA's Mussel Watch years ago, collecting samples, and then they discontinued that. And I'm looking around at their website now, and it's hard to see, but I don't see recent data from Alaska. And I don't know if any of this data gets sent to NCOS, NOAA, for inclusion in Mussel Watch, or if you know anything about the status of NOAA's Mussel Watch program in Alaska.
I do know a little bit, and I feel like Danielle knows a little bit too, so we could probably share this question. Mussel Watch is still happening. That's actually where the mussel component of LTEMP was based off, so they're kind of parallel studies, so we could compare across. So when I say we have low hydrocarbon levels in mussels, that is actually compared to the most recent Mussel Watch report, which was published just a few years ago and did include mussels from Alaska. We were part of Mussel Watch this year. They asked for some samples from their reference site near Mineral Creek, I want to say, between Mineral and Gold. So we had to collect an exorbitant amount of mussels to be part of their study, because not only do they look at hydrocarbons, but they also look at, I want to say, metals, maybe some more emerging contaminants like PFAS or yeah. They look at more things in the mussels than we do.
So have you looked at, I mean, Mussel Watch is a long-standing, they describe it as the nation's longest standing biomonitoring program. Have you looked at that historical data in Alaska in the context of the data you're collecting?
I have, but not recently, so it's not off the top of my head. But I do know, for example, that the mussels in Skagway Harbor were quite a bit hotter than our mussels when it comes to hydrocarbons. But I haven't looked extensively at comparing them to see if, levels have tracked the entire time with our LTEMP mussels.
Thank you.
I just add that we're on the distribution list for the results from this year's Mussel Watch sampling in Prince William Sound. So when we have those, we'll share them with Morgan and we can see. And it's pretty cool that LTEMP was based off of Mussel Watch, because a lot of our sites overlap. So they're sampling at Knowles Head and Gold Creek and a lot of the other similar sites.
Dave Jenka.
Thank you. Just wanted to backtrack a little bit on the lingering oil. I know that latest report by Ron Hines, he's retired from NOAA, and he got that through Exxon Valdez Oil Spill Trustee Council and ran it through Sitka Sound Science Center to do that survey. And I know in there he mentions how lingering oil is locked in the sediments. People should just leave it alone. Don't go digging these pits. I'm sorry, I'm not going to leave it alone. And there are, and he just makes it sound like there's nothing else that's out there that would disturb it, which I really disagree with. Haven't written him on it, but if you know Ron, I don't really want to criticize him for anything. But, some of the stuff is very shallow in areas with mussels, and otters are out there digging in it. Pycnopodia sea stars are coming back, and they dig for clams and dig quite deep in the sediments and expose that. I've seen bears come down and dig around in mussel beds, and large storm events. I mean, some of these wind events we had this winter could easily start tumbling things around a lot differently, because they're 10, 20-year wind events. And I'm sure we have some 100-year events coming up at some point that is going to change these things around. So I kind of just wanted to clarify, there are things that can expose that. And thank you.
Thank you, Dave. Seeing no further questions, great report. I'm impressed.